Thank you for the introduction, Chairman. And I'm Masaaki Kotera from Tokyo Disneyland, from Tokyo Institute of Technology of Japan. And it's my great pleasure to have a talk here in this conference. Okay, so my main interest is about metabolic pathway, and for which a lot of researchers have already been studying. And most of them are concerned about difference pathway that consists of 5,000 metabolites with known reactions. Let's say that these circles, black circles, represent metabolites, and these lines, edges, represent enzymatic reactions. And remember that difference pathway only considers the chemical transformation of metabolic compounds. And if we are interested in particular organism, particular species, then we can consider species-specific pathway, and that is a subset of reference pathway for which particular species are supposed to have. And then when we have some more detailed information, such as reaction flux, then we are able to do some flux analysis like this, as indicated in the slide. But our interest is something different. We are interested in the orphan metabolites. That is, we know about the structures of a lot of metabolites, but most of these metabolites are not known how they are synthesized or degraded, meaning that orphan metabolites is, well, we know about structures, but we don't know about the reactions. There are so many such orphan metabolites, so the reference pathways is something like this, surrounded by a lot of orphan metabolites. The number of metabolites is estimated to be more than 1 million. Okay, so that is the current situation for the total metabolome, that is untargeted metabolome, and our interest is about how to construct de novo pathway reconstruction study. Yeah, so it is the estimation of prediction of putative reactions among the orphan metabolites. Yes, that is the aim of our study. So let me explain about the concept of enzymatic reaction likeness. So when two pair of compounds are given like this, with connected by an arrow, then does this look like an enzymatic reaction? Well, what do you think? And here's another example. When a pair of compound is given, then how about this? Then does this look like an enzymatic reaction? Well, in fact, the first example is a positive example, meaning that we know that an enzyme can catalyze this reaction. And the second thing is a negative example, meaning that no one knows if any of the enzyme can catalyze this reaction. All right, so the problem of enzymatic reaction is that is the concern about de novo pathway reconstructions. And the main problem is to distinguish these positive examples and negative examples. So we have been tackling on this matter for many years, and the most recent one is presented in ISMB 2015, that is the last year's conference, and that is about reaction prediction from chemical structure. And basic idea of this study is to build graph alignment-based feature vector and train SVM. And advantage is the ability to improve the accuracy and extract biologically meaningful features. So let's consider that we are now interested in the enzymatic reaction likeness between these compounds. And the first thing to do is to make a molecular graph like this, a labeled graph of that represents a molecule. And we make a chemical structure alignment like this, and that is atom-to-atom mapping that extracts the conserved substructure and the not conserved substructure like this, and that can generate the alignment-based feature vector for a compound pair like this. And I skipped the detail about this vector, but it represents which chemical substructure are preserved and which bonds are eliminated or generated. And once we get this feature vector for a compound pair, then SPM and any other classifier is available to classify this compound pair for positive or negative examples in terms of enzymatic reaction likeness. So let me put this problem another way. Previous studies is about reaction prediction between orphan metabolite compounds. So let's say that these circles represent orphan metabolites for which we know about chemical structures, but we don't know about the reactions. Okay, so the previous study is about predicting reactions between the orphan metabolites. So that is these dotted lines represent the putative reactions between the orphan metabolites. And that was the previous studies. And this study is something different. Well, this study is about the prediction of reactions as well as the corresponding enzymatic enzyme orthologs. And here the dotted lines represent putative reactions, and the rectangles represent enzymes that are responsible for the putative reactions. All right, and then the main point is how can we predict enzymes for these putative reactions? That is the point of this study. All right, so this before explaining the detail of this concept, I'd like to ask a question. Do you know what is the most important species in the world? Human? No, it's a Mickey Mouse. This is the most important species in the world, right? And do you know what is the second important species? Yeah, it's Donald Duck. And humans are less important than that. Well, let's say we consider a known reaction like that, and we know Mickey Mouse very well, and then we know there is a verified function. Yeah, so gene 1 have a verified function about catalyzing this known reaction 1. And when we want to know less important species like this, then it's very costly to do some experiments. So then this is a place to where the bioinformatics gets power like this. We know that some homologies between gene 1, 2, and 3. Then we can say that these genes forms enzyme ortholog. In such a case, we can predict that these enzyme orthologs have putative function to catalyze this known reaction. Yeah, this is a very basic strategy in bioinformatics, and we know that this is very powerful for the genome annotation. Yeah, but this is my interest is something a little bit different than that. Putative or unknown reaction, that is my main interest. And there are some more species other than that that possibly have this putative reaction, such as swan, dolphin. Yeah, so when there is such organism, such species that are supposed to have putative reaction, but this is just, remember that this is just a putative reaction, so we don't know, we have not verified this reaction, and so the problem is how to get annotation on these putative reactions. So the best thing we can do is to search for the similar reaction to the putative reaction and get a verified function with one of those genes. Then we search for the homology and get an enzyme ortholog. Then we are ready to get a putative reaction, a putative function to get to catalyze the putative reaction. Yeah, this is the most important thing to do to explain our study this time. Yeah. So this is the aim of this study. The first aim is the prediction of the associated enzyme orthologs catalyzing the putative reactions. And the second thing is the extraction of the chemical transformation patterns that are characteristic of each enzyme ortholog. All right, so we used 15,000 metabolic compounds in check database, and the number of all possible compound pairs is 246 million. and 7,000 substrate product pairs associated with at least one of the 2,500 enzyme orthologs were used in this study, and positive and negative examples were defined for each enzyme ortholog. And here's the method. We constructed a specific linear classifier for each m numbers of enzyme orthologs, and this is the equation to explain this strategy, where W represents a weight vector for compound feature vector of a compound pair, and phi represents the alignment-based feature of the compound pair that for which I explained it in an earlier slide. And the problem is there are little information on non-metabolic substrate product pairs for many enzymes. So we solved this problem by using joint learning JL of weight vectors of M classifiers with enzyme sequence similarity. And we made a jointly learned weight vectors so as to force weight vectors to be similar for similar enzyme orthologs. By solving this minimization problem that consists of R, logistic loss of all pairs of compounds, and TR for the L2 regularization. And we incorporated the sequence similarities using the similarity matrix S for the between that represents the similarity between the enzyme orthologs, and the W, the weight vectors for the compound pair feature vector. Yeah. And as a result, we found that the proposed JL method outperformed the nearest neighbor method in terms of higher AUC scores. And in here, nearest neighbor method is based on the similarity search that is the most popular in the reaction classification in the previous studies, including mine. Yeah. And in this picture, this horizontal axis represents the enzyme orthologs, and the vertical axis represents AUC scores, and left panel and right panel shows the results from NN and JN. And as shown clearly, in many cases, JL outperformed JNN method. And we also made some investigation and found that AUC scores for enzyme orthologs were improved by JL. And here, especially with the low degree, where the degree means the number of positive examples for each enzyme ortholog, and which are represented as a horizontal axis here, and the vertical axis represent AUC scores, and NN and JL results are represented in the left and right panel as well, respectively. And it is clearly shown that JL performed better, and especially with the low degree of the enzymes. And this enzyme are also proposed able to extract the the common characteristics, important characteristics of chemical transformation patterns as highly weighted features. So this is one of the examples of an ortholog that catalyzes two reactions as this. And as you can see from this picture, we can easily extract the common characteristics of the chemical transformation like this, as indicated in the red circles. And that can be, well, our proposed method successfully extracted this kind of common characteristics as highly weighted features like this way. Each of the chemical structures are represented as graph structure, molecular graph, labeled graph, which are subjected to a chemical structure alignment, that is atom to atom mapping like this, and then we got the feature vector like this. And the highly weighted features were found to be corresponding to enzyme-specific chemical transformation patterns as indicated in these red things in this picture. And we further investigated the result, and we also found that in many cases, EC numbers were different, but reactions and sequences are similar to each other. So this example is taken from the previous slide, which is reactions that catalyzed by an ortholog that belongs to this EC number. And here's another example of that comes from the different EC number and different ortholog, but the reactions something similar, based well in terms of the common chemical structure transformations, right? So and all right, so these reactions are catalyzed by different enzyme orthologs, but it has something similar in terms of reaction characteristics, and despite of the EC numbers, different EC numbers, and not only that, these enzymes belongs to the different orthologs, but in terms of the Smith-Waterman scores of the protein sequences, in fact, they are sufficiently similar to each other, and this details is explained in our paper. This is not the only case. There's another case where EC numbers were different, but E reactions and sequences are similar, and this is another example that comes from the different ortholog and different EC numbers, but the reactions are where substrates look very similar to each other, right? And in fact, our result have the ability to get the Nobel prediction, and in fact, the Nobel prediction predicted by this ortholog and this ortholog were identical. And that, I think that is very natural because the substrates are very similar to each other, and not only that, the chemical structure, chemical transformation patterns are also similar to each other, as indicated in this red thing. Yeah, and also, although they belong to the different ortholog, but in fact, the protein sequences are very similar to each other in terms of the Smith-Waterman scores, as explained in our paper. All right, so in summary, the proposed method enables to predict putative reactions and the corresponding enzyme orthologs. And it was possible to extract enzyme-specific chemical transformation patterns. And the reaction similarity was also supported by protein similarity. Okay, so this is the end of my talk, and thank you very much for your attention.